This lecture will cover processing blood and other specimens. So again, I'm going to put my motto out there. The result is only as good as the specimen. So if your specimen is not properly collected, labeled, stored, processed, then that's going to cause errors when you go to report a result. So making sure that that specimen is good is going to make sure that your results are good as well. So you need to know what to consider when you're looking for what is a good specimen. So you need to be able to look at things like the specimen requirements for specific tests, understanding which samples or tests goes to which department, any kind of factors that can affect that test. What should you consider when you're looking for a specimen to be unacceptable? What should you do to preserve and store those specimens? What should you do to make sure that you're not having any kind of contamination from your environment or workstation? What the specimen is collected in is very important. So you have to look at your specimen collection containers. So when you're working in the lab, you've got to be familiar with the different variety of specimen collection containers that are out there and available to you. Of course, we know test tubes. There are a variety of test tubes, but there are also other specimens that you'll get into the lab, and you got to be aware of those containers that they're collected in and if it's the appropriate container. So with your blood collection for blood cultures, it needs to go into a blood collection or blood culture bottle. That's going to be dependent on the machine that the facility uses. So these can look a little different, but beyond just the bottle itself, it can be further divided into a aerobic versus an anaerobic blood collection system. So here at the top in this corner, you've got a few varieties of blood collection containers for blood cultures. So you have an aerobic one and anaerobic one. This has to do with bacteria that can live in an oxygen-rich environment versus ones that cannot. The AFB blood culture bottle is looking for acid-fast bacteria. So your bacteria require different things in order to survive. So making sure you collect in the proper bottles is going to make sure you get the best chances overall of detecting what could potentially be making that patient sick. And then you have some different plastic collection containers depending on what you're doing that are sterile. So sputum collection cups, urine collection cups, stool collection cups. Down here at the bottom, you're looking at a specimen collection container for virology. And so it has a solution at the bottom here that helps to ensure that those viruses are maintained long enough for you to test them. Looking at things like catheters, specimen swabs. So depending on what you're culturing, you could get a specimen swab, or those specimen swabs may have something in it to kind of help to prevent that swab from drying out or to prevent it from being exposed to oxygen. And so there are a variety of different collection swabs depending on what you're looking for, what kind of infection you're suspecting. There are wire swabs out there. There are plastic swabs. The swab tips themselves can actually be a little bit different. They're made out of different things. So depending on the particular test method, there are some swabs that you can't use. There are some tests that may require just a rayon swab and not a cotton swab. And then of course you have your variety of collection tubes that you could potentially have to use. When collecting a blood specimen, you've got to again know what the test is that was ordered and know what the specimen type is for that particular test. And then in addition to that, making sure you get the correct tube, you also need to ensure that you collect the tubes in a specific order. Now again, I think I've mentioned in another lecture where your potassium tube, or your EDTA tube, which is this purple tube here, it has actually got potassium in it. That's part of what prevents that clotting mechanism from happening. So if you were to collect your chemistry tubes, which are more of in this section right here, if you were to collect a chemistry tube and put that chemistry tube after your EDTA tube, then it could be possible that that EDTA potassium that's in that tube could get on your needle and contaminate your chemistry specimen. So you have to look at what your order of draw is and memorize this order of draw, because you don't want anything to come into play that could affect your test. In addition to the order of draw, there may be requirements of how many times you need to actually invert that tube to mix up that anticoagulant. So as soon as you do your venal puncture and you're switching out your tubes to go from one tube to the next, as you're pulling that tube off, you immediately want to do several inversions and place that tube down. So as you're moving your arm to place that tube down, secure that tube somewhere so that it doesn't roll off and drop. Then you're mixing that tube as you're reaching for your next tube. So as you're swapping those out, if you keep in mind to mix those tubes as you swap them out, it's going to kind of help you. And then of course most people, after they've done that collection and they've got all their tubes done and they're holding the gauze or bandage or whatever on that patient's puncture site, they'll pick those tubes up and do another inversion or two just to make sure that everything's mixed properly. If your specimens are not properly mixed, then that anticoagulant may not mix in that blood and you could potentially get a clot in that tube, which is going to cause problems. One of the key things that you need to learn, and the sooner that you learn it, the better, and I do quiz my students on this a lot. What is serum? What is plasma? So essentially they do look the same. They're both that clear portion of fluid that is above those cells once you spin a tube down. So to know the difference between serum and plasma, you got to understand the specimen tube type that you're collecting that sample in. So serum is going to be the fluid portion that remains after your specimen has clotted and then you've spun it down. So when that specimen clots, you can see here in this bottom image where that specimen has clotted and you've got all of that specimen kind of maintained in like one big gel clump right here at the bottom of this tube. So when you turn this tube on its side, it does not flow down. You may have a little bit of liquid that flows down where that plasma is already trying to separate out, but for the most part you're just going to have one big clot. And so when you spin that tube down, it's essentially going to look the same. So your serum tubes are your red tubes, and it could be a red tube or a gold tube, but essentially that little rubber stopper at the top is going to be red. Now there are some variations in your serum tubes. You could have some that have that little gel separator in it. This is not one of those. So this will be your gold tubes. They're kind of a yellow color, but most people refer to them as a gold tube. They're going to have a little layer of gel in them that you'll see in there. And then your gel separator will also be indicated by what they call a tiger top tube. So a tiger top tube will actually have a little bit of like grayish, darkish black area on that actual rubber stop tube. And then you will have that little layer of gel in the center as well. Your gold tubes actually have something in it that kind of helps activate that clot, so that's why they're a little bit different. Now your plasma, on the other hand, that's the fluid portion with your where your anticoagulant has prevented it from clotting, so your clotting factors are still present in that plasma. Now with your serum, that clot has formed, which means any of your clotting factors that were needed to cause that clot to take place, they've all been used up, so they're no longer in that liquid portion. Whereas your plasma, there's something in it that prevents that clotting from happening, so therefore there's no clot in that tube. So when you spin it down, your clotting factors will actually remain in that clear liquid portion. So all of this portion right here still has those clotting factors. So there's where you need to know the difference between serum and plasma. Plasma has clotting factors, serum does not. It's also important to know where your specimens go. So to do that, you've kind of got to understand the different areas of the laboratory. So your chemistry department is basically going to be various analytes, your glucose, your potassium, calcium, all those things like that. Your metabolites, you have different drugs of abuse, different therapeutic drugs. The chemistry department itself has a lot of tests, a lot of your enzymes, checking your kidney functions, your liver functions. So most of the time they're going to take your green and gold tubes, your red tubes, immunohematology or transfusion services, also known as blood bank. That's where the department that's going to be responsible for making sure the testing is done for potential transfusion of various blood products and components. So we're checking your antigen and antibody responses. And so that department's going to use EDTA blood, sometimes serum, but mostly it's going to be your EDTA blood for your red cells and your plasma. Hematology is going to look at the cellular components of blood. So with your hematology department, you're using whole blood because you're looking at the red cells in their normal environment where they're free floating. So you don't want this tube spun down. Hemostasis is going to look at your ability to coagulate, your ability to form a clot. Sometimes this is such a small area, it's kind of incorporated into the hematology or the chemistry department. But they're going to have the citrate, that blue tube, that light blue tube is what your hemostasis department is going to use. Microbiology is basically going to be focusing on your pathogens, what can infect the human system. So you're going to get more of your culture type specimens, your culture swabs, your blood cultures, your viral detection, your mycology detection, your parasite detection, stool specimens, things like that. Immunology, serology is usually incorporated into another area of the laboratory, but again, this is going to look for your antigen antibody response, whereas blood bank looks at it from an actual transfusion level. Your immunology, serology is going to look at it more at a immunity level, so checking for your body's immune response to particular stimulants. And then your urinalysis and body fluids, again, it's kind of incorporated into either hematology or chemistry, but that department is going to be looking more of analyzing any sort of urine specimen or body fluid specimen like your semen or your joint fluids. So again, depending on the department will depend on what particular specimen you would be using for that department. And this right here is kind of a condensed list. There are some tests that may require something a little bit outside of this realm, but for the most part, this is kind of what you're going to see as far as the specimen that's delivered to which department. Now, deciding whether or not to spin a specimen, that's going to be dependent on that particular test that you're doing and what the policy is on whether or not you should spin that specimen. So if you do need to spin a specimen, it could mean that you might have to do some adjustments with your centrifuge. You have certain specimens that may be required to spin for 10 minutes, where something else may be 5 minutes. The RPMs on that centrifuge may need to be adjusted. You may have something that has a harder spin, something that has a softer spin, depending on that test. So you need to make sure that whatever you're doing, if you're the person that's responsible for prepping that specimen, that you're spinning that specimen at the appropriate time and speed for that particular test. If not, you need to make adjustments to your centrifuge. Once you've centrifuged it, then you're going to look at it to see if is it an acceptable specimen. So we're going to be looking at things like the hemolysis and lipemia. Your specimen type may require that you look to make sure it's not clotted. So for example, in hematology, if you're required to use whole blood samples, then if there's a small clot in that tube, it can actually affect your results. So you need to kind of make sure that that sample's not clotted. In the same same aspect, if it's a sample that needs to be spun, then you need to make sure before you spin that sample, if it is a serum tube, that that clot has actually formed, because if that clot is not fully formed, when you go to spin that sample, you're going to have a mess on your hands. You're going to have this little gel clot that forms because you didn't allow those clotting factors to finish their job. So they actually continue it while they're in the centrifuge, and then your plasma itself, your serum itself will actually clot, and you'll have a clot above your red cells. So some things that can kind of come up that could cause you to reject a specimen or consider it unacceptable would be that someone sent you the improper tube for that particular test. Your sample came in with a clot in it. So for example, if you get a clot in your hematology tube, in that lavender tube, then what's going to happen is that clot's going to actually cause those red cells to your red cells, your platelets, white blood cells to get hung up in that clot. So when you're trying to do a count on the number of red cells or platelets or white blood cells in a specimen, if they're half contained inside of that clot, then you're not getting an accurate number. So if you suspect that there's a clot in that tube and you check it, there is a clot in it, then you're going to have to ask for a new specimen. A hemolyzed specimen. So again, these samples here at the end are considered a hemolyzed specimen. So you can kind of see that reddish tint to it in that specimen where it's hemolyzed. An icteric specimen. This last one here on the end, it's actually hemolysis, but it's kind of got a little bit of that icteric gold appearance to it. But if something is icteric, then it's got more of a darker brown amber color to it. And this is because that patient has got some kind of bilirubin that's spilling over into their circulation, and so it's going to kind of cause that dark look to that specimen. So this is usually going to be somebody with liver problems, or potentially, if an infant is born premature and their liver's not fully mature to help remove any of that waste from the blood, then it's going to kind of build up, and this is where you start seeing babies that might be a little bit jaundiced, maybe have that little bit of tanner looking skin, or that yellow color to the eyes, and then those babies have to go under those UV lights to help break down that bilirubin. So bilirubin, when exposed to light, will actually start to break down. So that's another consideration, is if a specimen is collected on an infant to do a bilirubin test, you've got to prevent that sample from being exposed to light. So putting it in something like a dark container to shield that specimen for light is very important, because if you're falsely breaking down those bilirubin particles inside that specimen due to light, and you test that specimen, you send it out, and it looks normal, but they're really elevated, then that infant's not going to get the treatment that they need. And what can happen over time is that bilirubin actually damages the brain. A specimen that is lipemic is going to kind of have a milky appearance to it. So these specimens here, these first three, they're actually lipemic and hemolyzed. You can see that little pink tinge to it. So those little fat particles that are in that specimen, you see this a lot if someone has ate a big breakfast. So they had eggs and bacon and sausage, and those chylomicrons will show up inside of that plasma because the body's not processed it yet and broken down that fat and used it. So you could potentially see something like this. Now there are patients that their cholesterol is just extremely high. They've got a lot of those triglycerides and things in their specimen, so you could see something like this. Well, what's going to happen with the lipemic specimen is it's going to interfere with any of your tests that use any sort of spectrophotometry through it, where it's actually trying to pass light through that specimen. Obviously, if you've got something in there that's that milky color, it's going to block that transmission of light. So therefore, it's going to interfere with that test result. So with a sample like this, it may need like an ultracentrifugation, where you actually spin it down You take out that plasma, put it in something separate, spin it down at an even higher speed to force out those lipid particles and those chylomicrons. Something else that you have to look for is if someone were to collect a tube for chemistry, or maybe they're performing a venopuncture and they realize, hey, I didn't get this tube, so they take another tube and pour it off into, let's say they take an EDTA tube and pour it into a chemistry tube. Well, that potassium that's in that EDTA tube is going to go into that chemistry sample, and then it's going to give you false results. So you will learn over time as you're observing a test within the laboratory. You're going to kind of start noticing when something might be contaminated with like an IV solution, or something like cross-contamination from anticoagulant in another tube. So again, this kind of further breaks down some of those things that you can encounter with a clotted specimen, a hemolyzed specimen, an icteric specimen, a lipemia, and any kind of cross-contamination from tubes. So keep these things in mind as possibilities of interfering and why they're going to interfere with your test method. So part of that QA process is making sure that you're able to track a specimen. So making sure they're logged is very important, because that helps you to track where that specimen integrity was kind of breached, or where the error actually happened. So when you collect a specimen, you need to make sure that you record the date and time, the person that collected it. Once it gets into the lab, it's usually logged who received it, so that you kind of know who went through that specimen prep process. The person that actually results out the test, that performs the test and results it out, that's usually documented somewhere. If you're in larger labs, most everything is done through a computer system and through barcodes. So when it comes in, it's barcoded and received. It's when you result it in the computer, you log in to result it, so it tracks you through your username and things like that. If you have to do anything like call a critical value or anything like that, documenting that call somewhere, whether it's in the computer system or some kind of handwritten call log, that is important. That's one of those things that an inspector is going to look at when they come and do an inspection of your laboratory, because that is a clear requirement that you have some way to track all of these things. So it could be done through your LIS, or your laboratory information system, so your computer system for your laboratory, or it could be some kind of manual handwritten log. Whatever the laboratory's method is, it has to be tracked. This is a clear regulation so that you can kind of troubleshoot if something happens and you have a problem along the way. Part of those regulations is also to make sure that you have a way for preserving and storing specimens. You got to have something written out of where the specimens are stored, what the temperature range is. You have to have proof that you've logged those temperatures daily, that you've ensured that those specimens were stored for the appropriate length of time, that they were disposed of when they needed to be. Your inspectors are going to look at all of these details and make sure that you've got something in place. When they're looking at your storage requirements, they're also going to make sure that you've checked those requirements on a periodic basis, and they're going to look for a log of that. They're going to make sure that you have kept it within those particular reference ranges. Most labs kind of establish those ranges based off of what the manufacturer says. And so that's something that whoever is setting up that QA policy has to look into. The typical refrigerator range is 4 to 8 degrees Celsius. However, you may have a manufacturer that says that this particular kit can only go from a 4 to 6 degrees Celsius range. So you may have to adjust your policy for your lab to be at a 4 to 6 degrees Celsius range versus a 4 to 8 degrees Celsius range. And there are some specimens that occasionally have to go through a chain of custody. So anything that goes through a chain of custody is usually because it could potentially result in some sort of medical, legal legal issue. You see this a lot with drug screens. So your drugs of abuse have to go through some sort of chain of custody. So if you go, for example, if you go for a job that requires a drug screen test as part of that pre-employment screening, then that drug screen is going to go through a chain of custody. And this is so that it is tracked from the time it's collected. It has a seal on it. That seal is not to be breached, because if it is, then that sample is considered invalid. If you were to apply for a job and they said that you had drugs of abuse in your system, but you want to dispute that because you know that you don't, then part of that process is for that facility to prove that those chain of custody steps were maintained. And so that means you got to have some sort of way of tracking where that specimen was handed off from one step to the next and everything that was done within those processes. So something like this, it could potentially happen. Sometimes you see this in automobile accidents where there's any sort of intoxication that's suspected. They may actually come in and require that specimen to be, your legal team would come in and do a subpoena for that specimen, and then they could potentially do a subpoena for whoever was involved in that specimen process. So again, making sure specimens are logged from the time they are collected through the time they are resulted in some way, even if it's not a legal chain of custody document, through your records within the lab, that specimen tracking log, whether it's an electronic or some sort of manual method, is very important, because if you ever get subpoenaed in, and yes, you could be subpoenaed in because you ran a blood alcohol test on a specimen of a patient that was involved in an automobile accident. And so if you are subpoenaed to court, they're going to ask you, what did you do when you got the specimen in your hand? How did you handle it? So you've got to be aware of these situations at all time and make sure that you're documenting everything and that you're doing your job properly, because you could get called into a court of law for a witness. Accidents do happen in the laboratory. You know, we're all the time spilling something. Maybe you're transferring a specimen from one tube to the next and you spill a little bit. This is going to contaminate your work surface. So you need to make sure that you're always keeping your work system disinfected. You don't want to be using dirty pipettes because you didn't properly dispose of it. You need to make sure that your area is clean before and after your shift. You cannot rely on others to do this. The same aspect when you come into the student lab. Make sure you clean your workstation. While you're not supposed to eat and drink in the lab, occasionally you may take your gloves off to switch up to get a new pair of gloves, and if you lay your hand on that counter in that steps in between, you could potentially pick up something that you don't want. So making sure, if you do notice that you spill something while you're working, make sure you clean it up immediately. You don't want to leave something sitting on the counter that could potentially cross-contaminate your specimens or cause an infection in you or your co-workers. A good example of making sure you decontaminate your work surfaces is your microbiology department. You are dealing with potential biohazardous type things that could cause an infection. When you're doing PCR testing, the patient that you work with is positive and you contaminate your work surface, and then your next patient behind that patient is negative, but you accidentally drop your swab on the counter while you're preparing your test, and you've contaminated your patient that's negative with a patient that's positive. So these are all things you got to keep in mind when you're working, is not to cross-contaminate, making sure that you're always keeping a clean, organized workspace.